Hi, my name is Sophia Kramer, and welcome to my Capstone project where I focus on cheese making. When deciding on a topic to do for my Capstone project, I knew I wanted to do something I was interested in, and cheese is one of my favorite foods, so I decided to do something involving cheese. At first, I was really interested in the rind of cheese and the differences in microbiota between the rind and the interior, but I found that this topic was a little too narrow for a full capstone project, so I decided to broaden it a little bit and explore cheese making in general, keeping in mind pathogens we don't want to grow and the microbiota that we do want to grow through the fermentation process. So with that, let's jump right into the project. So first, just copy and paste the Google Drive folder into your web browser. And once you launch that into your web browser, you're going to see a Google Drive folder with my primary research article, my presentation that I use for the oral presentation, and then all of these little subfolders within the folder that we're going to dive right into right now. We are going to start our tour by looking at the types of cheeses folder, so go ahead and open that one. Then I really wanted to look at what kinds of cheeses there are out there. So open up the types of cheeses, and this really just showed me that there are so many different kinds of cheeses. There's 7 varieties described here, and the article mainly goes into the culinary science behind cheese, looking primarily at how to handle, store, serve, and cook cheeses. So after reading this article, I wanted to dive deeper into the diversity of cheese, which is the second PDF found in this folder, and this article is a more technical look at classification schemes for cheese. The article sorts the cheese based on texture, method of coagulation, ripening indices, and then it even goes into a very deep qualitative evaluation of each of the varieties of cheeses at the very end. After reading about the varieties of cheese, I wanted to see how cheese is made. So let's return back to the home folder and continue our tour with the folder titled Cheese Making. First, let's look at the basics and details of cheese production. This article gives a brief overview of the cheese making process. It describes how the addition of lactic acid bacteria to milk allows for fermentation to take place, which causes milk to sour. This allows for the separation of curds and whey, which is basically how cheese is made. The article even includes a diagram of fermentation, very similar to what we have seen in class. The lactic acid bacteria described in the article are called cultures, and I wanted to find more about them in regards to cheese making, which leads us to our next article. The other article in this folder is all about, you guessed it, cheese cultures. It starts by describing the function of starter cultures. The addition of lactic acid bacteria or yeast provide lactic acid via fermentation, which decreases the pH, allowing the milk to turn into cheese. The article says that choosing the right culture comes down to the rate of acid made, predictable characteristics of the cheese produced, and the absence of pathogens during production. Finally, the article mentions how bacteriophages can interrupt the culture from doing its job, which makes sense given what we have learned in class regarding phages. After being exposed to the idea of having bacteria in our food, I really wanted to dive into the world of microorganisms in cheese. So let's go back to the home folder and continue our tour with the folder titled Microbiota of Cheese. First, let's open up the Microbial Diversity of 12 French Cheeses article. This experiment analyzed 12 different varieties of French cheeses on a 16S rRNA level. The study found that 76 different bacteria and 46 different fungal phenotypes were identified across all cheeses. Cheeses that were made in the same factory tended to have similar microbial populations, which makes sense. The largest differences actually occurred between the core and the rinds of the cheeses, which made me want to do more research on the difference between the core and the rind. Let's take a look at our next article in this folder, titled Microbial Communities of a Variety of Cheeses. This article in particular takes a look at the differences in microbiota of the different cheese types of provolone, Swiss, and cheddar, as well as the location of rind and core. Staphylococcus was found to be the most abundant in provolone and Swiss, while cheddar contained mostly lactococcus. The rinds of all the cheeses proved to be much more microbially diverse, which makes sense, as this is the part of the cheese that is exposed to the air during the ripening process. These articles made me realize that there are so many beneficial microorganisms in cheese, but what about the bad ones? Well, Let's move on to the folder within this folder titled Pathogens in Cheese. This study used cheese that was on the market and tested them for the presence of pathogens. Out of all the pathogens that were tested, Listeria monocytogenes was found in 5 out of the 12 samples. After much analysis, the article ends by stating that Listeria is the main pathogen of concern in cheese. So I had my answer. But this made me think, why Listeria? Well, let's go back to the home folder and continue our tour in the folder titled Listeria monocytogenes. First, let's click on the Overview of Listeria article. This guide provides a lot of great information about Listeria, such as the common characteristics, exposures, at-risk populations, and survival of the bacteria, among a lot of other interesting information. The chart says that the organism is capable of growing at low temperatures under salty conditions, which makes complete sense as to why it's such a problem in cheese. Another piece of information I found interesting was that the ingestion of Listeria causes a disease called listeriosis, having a mortality rate of 10 to 30%. That's really high. Well, let's look at the second article in this folder to learn more about listeriosis. Alright, let's see what listeriosis is all about. The article first talks about listeriosis infections in animals, which is not really on topic, so let's scroll down to page 5. There, infections in humans. This starts with the fact that the incubation period is anywhere from 2 weeks to 2 months. Symptoms of the disease range from fever and diarrhea to skin rashes. Pregnant women are at most risk to the disease and are warned to stay away from problematic foods such as uncooked meats and cheeses made from unpasteurized milk. This got me thinking about the differences between raw and pasteurized milk. If only we had another article. Well, let's go back to the homepage and see what we can find. Hey, look at that folder titled Types of Milk Used. Let's check out the first document called Dangers of Raw Milk. It looks like raw milk is any milk that has not gone through the process of pasteurization, which kills harmful bacteria. Let's scroll down a little bit. Wow, these facts about pasteurization seem pretty great. It doesn't really affect the nutritional value of milk, and it can actually save lives. Let's go back to the folder and click on the other article to see what that has to say about raw milk. This article also seems to say that raw milk can contain harmful pathogens. It looks like Listeria monocytogenes is the most prevalent in raw milk out of all the pathogens listed here. We know what that bacteria can do to us. The rest of the article goes on to debunk all myths of how beneficial raw milk is. The conclusion states that there is no nutritional or health benefits to drinking raw milk and it should be avoided altogether given the pathogenic concerns. I wonder how pasteurization changes raw milk from being full of pathogens to being food safe. Let's go back to the folder and click on the pasteurization folder to learn more. Milk pasteurization process. Here we go. It says here that pasteurization is when milk is chilled and then heated to 161 degrees Fahrenheit and then held for at least 15 seconds. Then it's quickly cooled to a chilly 39 degrees Fahrenheit. This process heats the milk up to kill all potential pathogens and bacteria that can make us sick. Even the Centers for Disease Control and Prevention says that this process is beneficial and does not affect the nutritional quality of milk. No wonder people drink and use this milk to make other dairy products like butter and cheese. I wonder how we can make cheese even better in the future. Let's go back to the home page and click on the folder titled Advancements in Cheese. This article shares how different sensory and quality mechanisms are being used to make our cheese be produced faster with better flavor and texture. It looks like the main technical challenge is that it would be hard to implement into the cheese industry because it's so established. Well, that's the end of our tour, but thanks for coming on it with me. The primary literature that I chose to focus on can be found in the Capstone folder right here, so feel free to follow along as I talk about it. The title of the article is Influence of Milk Microbiota on Listeria monocytogenes Survival During Cheese Ripening. The authors are also listed below, but let's dive right into the introduction. The introduction begins by stating that there's a surplus of raw milk right now with an increased demand for cheese. It also dives into the food safety concerns around cheese as they can harbor bacteria such as Listeria monocytogenes, Escherichia coli, Staphylococcus aureus, and Salmonella. They also found a study in which there were coliforms that were reported in some cheese located in the United States. The next part of the introduction focuses just on Listeria monocytogenes, which is the biggest pathogen of concern in cheese, and we already found this out with the pathogen in cheese article in the Capstone project folder. Listeria monocytogenes is a zoonoic, gram-positive, facultative anaerobe, and it causes listeriosis, which actually has a 20 to 30% mortality rate, making it extremely dangerous. We can also remember this mortality rate from the Capstone folder in the listeriosis file. This bacteria prefers to survive in acidic conditions with high salt and at low temperatures. It really depends on the strain for the specifics, but these are the general environments that Listeria loves to thrive in. Because cheese is acidic, has a high salt content, and is normally stored at low temperatures, Listeria becomes a problem for the production and storage of cheese. The introduction ends with the objective of the study, which is to investigate the ability of different Listeria monocytogenes strains to survive in cheese made from raw or pasteurized milk, and to reveal the effects of milk microbiota on the survival of Listeria monocytogenes. Moving on to the material and methods, first we're going to talk about the inoculum prep, which is where 3 strands of Listeria monocytogenes, the 3 are listed there, were contained from animal and human subjects. And all 3 of these strands were inoculated into tryptic soy broth with 0.6% yeast extract. These 3 tubes were incubated at 30 degrees Celsius for 24 hours. After the 24 hours were up, 0.1 milliliters of the TSB were inoculated in 10 more milliliters of the TSB with yeast extract, and these were again incubated at 30 degrees Celsius for 24 hours. Then the subcultures were taken and centrifuged for 1912 per gram at 4 degrees Celsius for 15 minutes. Then they were washed twice with phosphate buffered saline. Because of the centrifugation, they turned into pellets at this point, and they were stored in phosphate buffered saline again, and then diluted with more saline until each test tube had about 5 to 6 log CFU per milliliter of the Listeria. Moving along to the cheddar cheese preparation, each of the 3 Listeria inoculum preps from before were put in either pasteurized milk or raw milk, and this made 6 different samples. Then each of the 6 samples were combined with 0.01% lactobacillus, which was the starter culture in this cheese making process, and it was also combined with lactose. Then each of the milk samples were incubated at 32 degrees Celsius for 60 minutes. After incubation, the samples were combined with 0.02% liquid animal rennet, and then they were left for 30 to 45 minutes in order to form curd. Once a nice curd was formed, they were cut into 2 centimeter by 2 centimeter cubes and then stirred, and they were slowly heated to 38 degrees Celsius via a 1 Celsius increase about every 5 minutes. Each of the samples were exposed to cheddaring, which is cutting and stacking the cheese, and then they were strained using a cheesecloth, and then 2% NaCl was added to each cheese sample. Then each sample of cheese was placed into a cheese mold, and then it was pressed at 25 degrees Celsius for 40 to 50 minutes. Then each cheese was repressed with 10 times the weight of the curd for an additional 12 hours, and after that it was stored at 13 to 15 degrees Celsius in order for the cheese to undergo the ripening process. The whole cheddar cheese preparation process was repeated twice in order to ensure proper replication. For microbial analysis, during the first 60 days of the ripening process, analysis was taken every 5 days, and during the 70 to 170 days of the ripening process, microbial analysis was taken every 10 days. Then 25-gram samples were cut off of the cheese blocks aseptically and put into filter bags with 50 milliliters of 0.1% buffered peptone water. Then, very similar to what we did in lab in class, they were homogenized using a mixer for 60 seconds. Cereal dilutions were then made from the bags with 0.1% buffered peptone water. 0.1 milliliters of the dilutions were plated on talcum agar, and the plates were incubated at 30 degrees Celsius for 48 hours. Then, for the enumeration of Listeria, the dilutions were also plated on Lactobacilli MRS agar, and those plates were incubated at 37 degrees Celsius for 24 hours. On the same days as the microbial analysis, water activity was also taken. These sample containers were filled up 70% of the way with the cheese samples, and the AQS31TC water activity meter was used for analysis. Next, to find and compare the microbiota of the raw and the pasteurized milk, next-generation sequencing analysis was done. The PowerSoil DNA isolation kit was used to extract the DNA from both the milks. A sequencing library was made via PCR using the Nextera XT index kit. Finally, to determine raw data counts, the Illumina system was used alongside many softwares. For statistical analysis, 2 replicates were used to strengthen data, as well as the software SAS, and a pairwise t-test with alpha of 0.05 was used for mean comparisons. So with that, let's look at some results. So these results here show the presence of the different strands of Listeria monocytogenes on both the raw and the pasteurized milk cheeses throughout the ripening process. As seen with the red circles in these graphs, both the raw milk and the pasteurized milk cheeses have a decreased CFU per gram of Listeria monocytogenes over the ripening period. Likewise, the water activity also decreased for the raw milk and pasteurized milk cheeses from about 0.97 in the beginning of the ripening process to about 0.75 in the end. When the comparison of the cell count on the day of sampling to the initial cell count became statistically different, it was judged as the first significant reduction. Looking at the raw milk cheese graph, this can be seen at the day 90, 50, and 80, depending on the strand. These can be seen with the blue circles. For the pasteurized milk cheese, this can be seen at day 55, 35, and 25, again depending on the strand and with the blue circles. These results insinuate that there is in fact a difference between the Listeria strands on the ability to survive in the created environments. The article also mentions that other studies actually agree with these results, as they found when they lowered the water activity of their samples, Listeria actually survives better in these conditions compared to other pathogens such as Staphylococcus aureus and Salmonella. Now let's take a look at the death rate of Listeria between the different strands. So first, looking at the raw milk cheese with the red circles on the graph, it took the strand SMFMSI6 about 120 days to fall below the detection limits, while the other 2 strands took a longer period of about 160 days. Now looking at the pasteurized milk cheese graph on the right, looking at all 3 of the strands, it took about 120 to 130 days to fall below the detection limits, as seen with the blue circle, which is a lot faster than the raw milk, at least for the 2 strands. As a result, we can say that the death rate of Listeria monocytogenes was slower in the raw milk cheese than in the pasteurized milk cheese during the ripening process. Now let's compare the microbiomes of the raw and the pasteurized milk. So using the next generation sequencing method, we found that the inverse Simpson values were both 0.98. This basically means that there's no difference between the two microbiomes on a statistical standpoint. However, there was a difference between the two TRAL1 values, and this measures the richness of microbial diversity. This insinuates that the raw milk has more microbial diversity, as the TRAL1 values for raw milk was 444 and the TRAL1 values for pasteurized milk was 354. This can also be seen by the red squares on the table, which shows that the raw milk contained a total of 5 phyla, that the pasteurized milk went undetected. When looking at the two milks' microdiversity on specific levels, the family, genus, and species levels all show that the raw milk had higher values for each of these categories. These can be seen with the blue squares. This further strengthens the idea that raw milk has a higher microbial diversity than pasteurized milk. Another idea to consider is the presence of organic acids in milk. Organic acids such as lactic acid, propionic acid, and acetic acid all offer antimicrobial benefits, and it's important to look at their concentrations in these two milks upon comparison. The organic acid producing bacteria of Lactobacillus, Lactococcus, and Streptobacillus are found in the phylum of Firmicutes, and this was found in a higher percentage in the pasteurized milk compared to the raw milk. The organic acid producing bacteria of Bifidobacterium and Rhozomonas is in the phylum of Proteobacteria. This is also at a higher percentage in pasteurized milk than raw milk, as seen in the red square. When comparing the percentages of organic acid producing bacteria for the two milks overall in comparison with the phylum, it can be seen that pasteurized milk has 6.4% organic acid producing bacteria and raw milk has 4.81%. As we learned in class and in the overview of Listeria monocytogenes article in the Capstone project folder, we found that Listeria monocytogenes, among other microbes, do not prefer acidic conditions to thrive. It was also found that the raw milk contained several harmful bacteria as indicated on the slide. This further agrees with the Dangers of Raw Milk article in the Capstone project folder that raw milk can cause several outbreaks if not treated correctly. The article ends by restating some of the major findings of the experiment, one of which is that Listeria monocytogenes survival really varies among the strain. Each strain really has its own unique preferences on parameters that the environments in which they enjoy to thrive in. Another finding is that the differences in microbial content of the milks, specifically with the presence of organic acid producing bacteria, really has an impact on the survival rate of Listeria monocytogene. The final words of the paper express that it's extremely important to know the dangers of raw milk cheese. In order to ensure that the cheese is really safe to consume, it's important to only eat cheese made from milk that has either undergone pasteurization or have added acid to inhibit the growth of Listeria monocytogenes. After analyzing the article, I do have a couple questions for my critical evaluation of the article. The first one being, why did the authors change the interval testing of cheese from 5 days to 10 days in the ripening process? If done 5 days throughout the ripening process from beginning to the end, there would be a little bit of a stronger data from the days of 70 days to 160 days. All strands of Listeria found their death phase in this latter time period, so I believe that it should have been 5 days interval testing from the beginning to the end. My second question is, were the samples brought to room temperature upon analysis? This could have had an interference on the presence of the 3 strands of Listeria, as each strain has unique conditions of which they can survive, and temperature can definitely be an impact on this. My last question is, was the milk being used cow's milk? Although not explicitly stated in the article, I do assume that this was only cow's milk being used. For my entire capstone project, that is what my project mostly aimed at. This is, however, an example of a limitation to the journal article, as they only tested cheddar cheese made from mostly cow's milk. This could have been expanded to other types of cheese, maybe made from other animal milk, which would provide more information of the idea of safety of cheese altogether. And with that, that is all I have for my oral presentation of my capstone project. I hope you learned a little bit more about cheese or cheese making today. Thank you so much for sitting through a 25-minute video presentation. I know it can be hard, but if you do have any questions at all, feel free to shoot me an email. I would be more than happy to answer your questions. Thanks, and have a great rest of your day.